Good morning, Preeti, Rajoshi. Welcome to the class. Priyanka, good morning. Yep. Shantanu, good morning. Okay, so let's begin. So down to the last class, last YouTube class for this month from my side, and that's Analytical Q A on Molecular Cell Biology Part 4. Okay, so if you go through all my classes that I have taken in this month, you will see that I discussed different types of analytical questions. 4 sessions you will get on different Q&A on experimental techniques, and 4 sessions on analytical Q&A on molecular biology. Okay, so 8 sessions you will get where I discuss some very important analytical conceptual questions. Okay, you just go and watch all those YouTube classes if you missed all those classes. Okay, so let's begin. So the last one from Molecular Cell Biology, Part 4. Master Suman Panda, educated on Unacademy Plus. Okay, so everyone, I just want to tell you that that's my profile. I think all of you know that. Okay, and I already have taken more than 140 special classes, and all those classes are free. Okay, you will get free recordings of all those classes in my profile. Okay, and so many different types of units, PYQ, model question answer sessions. Okay, revision classes I discussed in last few months. Okay, you just go through all those free recordings. Okay, and these are some free courses that I completed, and 14 plus courses I completed till date. 14 exactly. And if you really want to get a detailed course, if you really want to study unit 1, I mean biochemistry, molecular biology, central dogma, gene regulation, cancer, immunology, okay, then biotechnology, all those experimental techniques, then you will have my class courses. You will get all those classes from my class courses. Okay, just go through it, and you will get so many different Q&A sessions, more than 1,000 questions, exactly, more than 1,000 questions I discussed in my class courses. Okay, both Part B and Part C. So it's a big deal. It's a big deal for you to go through these class courses with subscription. Okay. See, and 3 class courses you will get for where actually I discussed all the techniques, methods in biology and chemistry. All these courses will be very helpful for all of you for all those exams, right? Okay, and see, these are all my completed class course, and if you really want to get all those courses, you have to subscribe. Priya, good morning. And right now I'm dealing with this particular batch course, so where I'm discussing 200 analytical questions for CSIR-NET. Okay, already I completed 8 sessions. Okay, so 5 more units to go. 8 is done, 5 more to come. So 200 top analytical Part C level questions I'm discussing in this particular class course. Okay, so if you really want to get all those things, you have to subscribe. Okay, and remember that that's my code, Suman P Live. Okay, whenever you subscribe, you just use this code, because these fee structures in the Plus subscription or in the Iconic subscription, these fee structures are only applicable when you will use this code Suman P Live. Remember that you will get 10% instant discount. Okay, while subscribing. Fine. So get on the subscription right away so that you can have all the completed ongoing one and the upcoming class courses. Okay, and all you have to do, you have to like and share all the videos, all my completed videos that I discussed already on YouTube. Okay, and subscribe to this particular channel, Unacademy CSIR-UGC NET. And don't forget to hit the bell icon so that you can get all the notification instantly. Okay, so I think we can start today's session, right? So first question for all of you, go for it. Okay, now so these type of questions you can get. I think you know that we have more than 20 different types of RNA still now discovered, still now reported. Okay, 20 different types of RNA. So you have to know their functions, their localization, okay, everything. Drop your answer. So all you have to do, whether you know the functions of all those RNAs, and you have to find out the correct matching. That's all. Devupriya, good morning. So can you see that, everyone? Can you see the column? Because you know it's not possible to zoom this particular column right away. Drop your answers. Very good morning to all of you. And let me tell you, I have one more session, special class from 1:20 p.m. today. That is also on Q&A, okay, that I am discussing. So be there. Okay, drop your answer, everyone. It's so easy. You just need to match. Go through it carefully. Done, everyone? Okay, so different types of answers I am getting, and let me tell you, see, first of all, let's start with siRNA and miRNA. Okay, so siRNA, what's the function of that? siRNA deals with turning off the gene, okay, by directing what? By directing selective degradation of mRNA, right? So B1, absolutely, that's siRNA does. Okay, what is miRNA? What is the function of miRNA? It deals with regulating the gene expression by blocking the translation of selective mRNA, right? Is it okay? That's what it is. So B1 C2, right? Okay. SNO-RNA. So what is the function of SNO-RNA? They process and modify rRNAs. Exactly. They process and modify, chemically modify ribosomal RNAs, rRNAs. Exactly. D4 and A3. SNRNAs function in a variety of processes, including splicing of pre-RNA. Right? Remember SNRPs, small nuclear ribonucleoproteins. Okay. Definitely. So option B is the right answer. This one. Okay. Fine. Next question. So you just have to remember all those things. You just need to study all the different types of RNA, and then only you can be able to get all the functions. Okay, you have to remember, right? So next question. Okay, so now this is something so incredible. I mean, this particular question is actually wonderful. Okay, you just go through this particular question. Let me tell you, even if you don't know, still you can answer this question. Just try to analyze it. Just try to analyze. This is a sucrose density fraction of different different ribosomal compartment. Okay, like 60As, 40As, 80As, and also polysome. And you have 3 different inhibitors which can inhibit translation. Okay, but their mode of action are different. That you have to point out, that which inhibitor can give you this type of sucrose density fraction of ribosomal compartments. Okay, go through the question carefully. I just want everyone to go through the question carefully so that everyone can give me the right answer. At this stage, you have to answer such questions. Okay, and I think these questions are easy to answer because you don't have to study anything. Just go through the question and go through the diagram, analyze it, and get the answer. As simple as that. Everyone, drop your answer. Shubham, Shantani, okay. All of you, go for it. Everyone, you take your time, okay, and give the right answer. Because all the 3 different inhibitors, they have different mode of action, different timing when translation is going on. So definitely you can expect different consequences, different outcomes, right? From the treatment of 3 inhibitors, right? Okay, so that's what it is. You have 3 inhibitors and 3 different outcomes. Just you have to match. That's all. Okay. Everyone? Priyanka, Devapriya, Shantani, Shubham, all of you. Okay. Okay. Let me tell you, actually all of you are right. Okay, the right answer has to be option A. And why not? Why not? Preeti, are you getting this? It has to be option A. See, Tanisha, it's not B. It can't be. It is only possible that we can have this right option. Okay, just go through it. See, if this inhibitor is a leaky inhibitor of translation, what is the meaning of it? Leaky inhibitor. Sometimes it can inhibit the translation, or sometimes there can be some leaky expression. Sometimes translation can occur. So leaky inhibitor, leaky. Okay, it's not, I mean the action of that inhibitor is not strong enough. Sometimes it's there, the action of sometimes the effect is there, sometimes the effect is not there. That is the meaning of this word, leaky. So in such case, how what kind of data you can expect? Don't you think so that you will get a small peak of 40 and 60? Definitely it will always remain there. Okay, You will have a ATS and also polysome. So you are getting polysome. The meaning is very simple. That the translation is definitely going on. That's why you are getting polysome, right? Where different ribosomes are actually sitting on the same mRNA, right? Polysome. Okay, so you are actually getting almost all those things: 40S, 60S, a peak of 80S, and also polysome. So definitely, translation is going on here. So this inhibitor is not that much, accurate or appropriate to inhibit translation, because you won't need an inhibitor. Okay, you don't want to rely on such inhibitors that have leaky expression after giving the treatment, okay, which can have a leaky expression. Sometimes they are working for translation, sometimes they are working against translation. So you can see, right? So A has to be A. I mean, one has to be A, definitely. Okay. Now, if your inhibitors arresting ribosomes at the initiation codon, okay, so your inhibitor is arresting ribosome at the initiation codon, what will happen? What will happen? So if the ribosome is already in the initiation codon, definitely your ribosome will contain the full subunit conformation. Okay, large subunit, small subunit, everything you will get. Otherwise, it won't be able to sit on the initiation codon. If your ribosome is in the initiation codon, you will get the full form. And ATS, we are talking about eukaryotes. Definitely, the eukaryotic ATS configuration is already being established when the ribosome will reach in the initiation size, right? So ATS, C, 60 and 40 will be there. So definitely 2 will be B, absolutely. And then after, there is no peak of polysome because translation has got the translation is got inhibited by this particular inhibitor, because it's already added the ribosome at the initiation codon. It won't be able to translocate. Okay, that's all. And obviously, if it inhibits the ribosome scanning, see, so this particular inhibitor won't allow ribosome to scan and to go through the sequence and find out the Kozak. I think all of you know that they have to find out the Kozak sequence, because depending on that, they can be able to predict where their start codon is, right? Kozak sequence. You have to search for it. They have to search for it. So if they inhibit ribosome scanning, if this particular inhibitor inhibits ribosome scanning, what will happen? What will happen? You don't even get the ATS construct. You will only get 40 and 60. Definitely, it will be there. 40 and 60 subunits will be always there in the cytoplasm, in the nucleoplasm. Definitely. Okay. Think about it. Cytoplasm, to be very specific, not nucleoplasm. Cytoplasm. Okay. 40S, 60S. Right. So after scanning, when they encounter with Kozak sequence and start codon, then only ATS configuration will be there. Okay. So 3 will be C. So option A. Okay. Understood, everyone? Tanisha, Preeti, you got this question? All okay? I think so. Next question. Okay, so I think we all know, right, that in prokaryotes the initiator tRNA is methionine, formyl methionine tRNA. We know that, formyl methionine tRNA. So one might have this question that okay, then every prokaryotic protein will start with methionine. Yeah, obviously, because the first amino acid is methionine. We know that. Or even one can question that first amino acid can be formilated methionine. Right? Okay, but it's not actually the case. And you have to study this particular region. Okay, and if you go through my past courses, I have discussed all those things in detail. Okay, step by step. I just completed the whole story. You just subscribe and get all the plus courses, because you know all those important topics I already discussed. Okay, if you really want to get a detailed preparation, just subscribe to my plus courses. Bushra, good morning. So everyone, drop your answer. Okay, so today I have another class from 1:20 p.m. Okay, it's a special class. Be there. Everyone, go through the question carefully and drop your answer. Good morning, Pallavi. You have to study central dogma well so that you can get these concepts. Okay, because these are some fundamental concepts, right? You can refresh it. I think it will be okay. Devapriya? Yeah, definitely. The options are options. Okay, A only, B C, E only, B D. Okay, so these are the options. Okay, so this is all about concepts. Okay, Priyanka, Preeti, Pallavi. I mean, you just go through my plus courses. Okay, you will get all the concepts down there. And when I will start talking about central dogma once again, you will definitely get all those concepts. Don't worry. Okay. Okay, so I think I have to explain this one. Let me tell you what happens. See, there's a particular enzyme called deformylase. Okay, deformylase removes the formyl group from the amino terminal methionine. Are you getting it? Deformylase removes the formyl group from the amino terminal methionine during translation or even after translation. Okay, so this is a take-home message. Just take it. Okay, so B is the right statement here. Absolutely. And another one is that aminopeptidase, another enzyme, a class of enzymes, right? Aminopeptidase. Okay, they remove the amino terminal methionine. Not often, not so often. I mean, they can do it almost every time also. Okay, aminopeptidase often remove the amino terminal methionine. Okay, and let me tell you something more. Okay, not only methionine, even aminopeptidase can cleave one or two more amino acids which is present there just by the side of methionine. Are you getting my point? Suppose this is methionine. Okay, this is second amino acid, this is third amino acid. So it is possible that aminopeptidase is cleaving this one, sometimes this one's too, or sometimes this one's too. So you're getting my point. So it's not possible to get a bacterial protein that's starting with methionine. Majority of them are not going to start with methionine because of this. And forget about the formyl group. It will be removed as soon as the translation is going on or just after the completion of the translation. Okay. So aminopeptides also take care of that amino terminal methionine. It will cleave it. Majority of the cases you won't get the methionine. Okay. So B and D. So this is a concept. Okay. We have the evidences. Okay, it's already standardized. Okay, so you need to study central dogma so that you can be able to understand all those things. Okay, so the complete story is this: not only methionine, even these aminopeptides can cleave one or two more amino acids sitting just after the methionine. It is cleaving this one. Suppose this is your aminopeptidase. Okay, it's chopping down from this side. Okay, so chopping this one out, this one and this one. That's it. Okay, so B and D. Option D is the right answer. Okay, so all of you need to study central dogma. Well, then only you can answer all those questions. Okay, next question. Just like another one from, I think from, translation. Go for it, quick. So once again, it's purely informative. If you go through my plus courses, definitely you'll be able to answer these questions. Okay. It's purely informative. You need to study. You should have all the basic concepts, fundamental concepts. Everyone, answer this question. You know, you will definitely can expect such questions in CSIR-UGC. Definitely, you will get such questions. So you just shape up your preparation in a way so that you can answer all the questions like this. Okay? Then absolutely. Thank you so much. All of you are right. C is the right answer. EIF4G and PABP promote the process through 5 to 3' interactions of mRNA. Exactly. Polybinding protein. Remember? Definitely. Okay. And PABP and EIF, initiation factor 4A, promotes this process. Definitely. So A and C, option 3. I mean option C. Definitely. This one. Okay. Absolutely. Okay. Got it, everyone? All okay? Nope, it is not A, it's C. Okay? Rajoshi, it's C. Okay, you need to study central dogma, otherwise it's not possible to answer such questions. You should have proper information, knowledge. Okay? And there are so many concepts right now available because in the last 10 years we got to know so many things, so many aspects of central dogma. Starting from stability, interactions, different proteins, okay, their sequence context, okay, and mRNA stability, turnover, so many things out there, okay. So you need to study. It's not just that you just take a random book and start studying central dogma. It's not that way. You won't be able to answer any questions, okay. There should be proper materials, okay, proper concepts, okay. So C is the right answer, definitely, okay. Fine. So you need to study, and if you really want to study all those chapters, everything, all you can do is just subscribe, okay, like, share this particular video, subscribe to this channel, hit the bell icon, and go through all my class courses from my app, okay. From Unacademy app, you just go through all the class courses. See, in this particular course I discuss central dogma, unit 3. You will get entire unit 3 in this particular class course. Okay, and so many, molecular biology, biochemistry, cancer, immunology, cell structure, function, biochemistry, all the techniques, biotechnology, everything I discussed, okay. And right now I'm dealing with this one, and all you have to do, you have to subscribe. So go for subscription right away, okay, just right away, just go for it. And Suman P Live, that's my code, okay. And let me tell you one more thing. These are the fee structure right now for plus subscription and for iconic subscription, okay. And there is an offer right now going on. If you go for 12-month subscription, see, if you go for 12-month subscription, okay, you will get 2 months subscription more extra, okay. So if you subscribe for 12-month subscription for 12 months, then you will get 2 months more, and also you can able 10% discount as usual. All you have to do, you have to use this code Suman P Live. That's all. So use this code Suman P Live. Remember, get 10% discount and also get 2 more months. So 14 months in the fee structure of 12 months. Okay, so go for this subscription right away so that you can have an overall preparation for CSIR, NET, GATE, DPT, ICMR, for all the exams. Okay, remember this code. That's it. Thank you so much for being there, and I'll be back again with a new series of special classes on YouTube, definitely. And today I have another special class from 1:20 p.m. Okay, so be there in that particular class. Okay, everyone, thank you so much, and don't forget to like this particular video, watch all my completed sessions on YouTube, share all those videos in different platforms. Okay, hit the bell icon and take the subscription. Okay, thank you so much, everyone. Take care. Thank you.